So Mickey is a professor of molecular pharmacology and has secondary appointments in neuroscience and pediatrics. He was originally from Jerusalem, Israel, and after contributing very serious service as a paratrooper in Israel, he came to the United States to do his higher education. He received and earned his BS and Ph.D. at the University of Rochester, and his Ph.D. was in neurobiology and anatomy. After doing postdoctoral work at Rochester, he moved on to an assistant professorship at Albany Medical College and rose to the rank of associate professor there. In 1994, he was recruited to Wake Forest Medical School in North Carolina, where he rose to the rank of professor. And in 2004, Mickey was recruited as the Stallman Chair of Neuroscience at Vanderbilt University. And during the course of his extensive research at Vanderbilt, he organized and directed some important facilities which he was responsible for, two funded by the National Institute of Environmental Health and Safety. One was a molecular toxicology center, and another was an important training program in environmental toxicology. So foreshadowing our forthcoming alliance with Montefiore, in 2013, Einstein and Montefiore and our department and a number of other departments at Einstein collectively were involved in recruiting Mickey to come here to Einstein. And as you may have gathered from what I've said so far, Mickey is an internationally recognized expert in toxicology and pharmacology. He has special expertise in neurotoxicology, and he has a major focus on heavy metals. And he really brings to Einstein a new area of research expertise that we haven't had in this institution and in our community. And his interests range from neurobiology, basic neurobiology, to neurotoxicology, mechanisms underlying Parkinson-like diseases, analysis of membrane transport systems, and utilization of C. elegans, for example, to elucidate mechanisms underlying neuronal degeneration. And he's also developed a recent interest in cancer biology. So many people within the Einstein community have points of contact and potential collaboration with Mickey, and a lot of that is already underway. In Mickey's early work, he focused on methyl mercury toxicity, and he made many discoveries. I'll just mention one or two. One was he discovered the carrier protein system that transports manganese, methyl mercury, into glial cells in the central nervous system. And then using cell culture and animal model systems, he and his colleagues found that methyl mercury disrupted mitochondrial ATP synthesis, generated reactive oxygen species, which in turn altered neurotransmitter transporters so that in changes in the astrocytes actually are then transmitted to neighboring neurons because you build up a lot of excitatory amino acids in the synapses, and this causes ultimately neural degeneration. He has a strong parallel interest in the cellular molecular toxicology of manganese, which we're going to hear about today. And among previous work, he and his colleagues characterized the transporter that is involved in accumulating high levels of manganese in astrocytes. And the consequences of this are changes in metabolism, gene expression, an inflammatory response, and changes in metal transporters and other transporters, neurotransmitter transporters. The result of this is there's a persistent accumulation, again, of excitatory amino acids in synapses and consequent neurodegeneration. So these are the kinds of contributions his lab has already made. Very recently, he's taken up an interest in using the molecular genetics of C. elegans to explore transporters, gene expression, signaling pathways that contribute to or protect against manganese toxicity. And in this system, he's found a very interesting relationship between interactions between manganese and dopamine that would as a consequence of which dopamine-containing neurons die. An important model system. So Mickey's work has been published in over 200 papers. He is constantly being invited to write many, many reviews. I'm not going to go into any details of that. You can look him up in the literature. He serves on numerous NIH panels. He's been a past chair of an NIH study section. He's currently on a major NIH study section. He's the editor-in-chief of BMC Pharmacology and Toxicology, and in his spare time, he's an associate editor on 7 other pharmacology toxicology journals. He does play a role in national public safety and public health. He's been an advisor to the federal government, to the FDA, to the National Academy of Sciences, to the EPA, and others regarding all kinds of issues in environmental toxicology. So there's an important, interesting role there. And he's been awarded a number of honors in his community. I'll just mention a couple. He is the past president of the neurotoxicology section of the Society of Toxicology. He was on the board of directors of the Academy of Toxicological Sciences. And in 2011, the Society of Toxicology awarded him the prestigious Merit Award for distinguished contributions to toxicology. So it's a pleasure to have Mickey join our community, and he's going to bring us up to date and review his work and bring us up to date on manganese toxicity. And Mickey.
Thank you, Charlie. I should have invited my mother. Okay, so I'm going to talk to you about some of the recent work that we've done over the last year, and as Charlie mentioned, I'll focus on manganese. So most of you are probably not familiar with manganese. I'll start with a brief introduction to manganese. I'll move on to talk about some of the work on the transporters that we're doing in different systems, and towards the end I'll try to bridge everything with some information on translational work that we've done in neonates, human neonates. So manganese is an essential metal. Unlike methylmercury, we all need it for normal function. It's absorbed in the gut, and as you'd expect from a metal that's essential, it's quite well regulated at the level of the gastrointestinal tract. The problem with most of the occupational exposures is that the manganese is absorbed through the lung, where we don't have homeostatic mechanisms that control as tightly the uptake of manganese. So basically on a molar basis, if one is to be exposed to the manganese by lung, the manganese tends to accumulate in different tissues, especially in the brain, at much higher concentrations. And once it's in the blood, manganese is transported on different proteins, different globulins. And as I said, it's essential, so it's basically distributed to all the cells. Now, the reason that we're concerned about manganese is that high levels of manganese exposure tend to heighten the risk for the development of a Parkinsonism-like disease. It's very similar to Parkinson's disease, and I'll show you a slide later to explain why it's so similar, but there are some distinctions between the two. One of the take-home messages is that manganese and iron compete for the same transporters, and I'll show you that as well. So that manganese accumulates in brain regions that are responsible basically for controlling movement, lobus pallidus, putamen, and areas similar to that. The populations that are at risk, I'll talk about parenteral nutrition. The reason for that is that manganese is just supplemented at very high concentrations in parenteral nutrition. Iron deficiency, as I mentioned, because of shared transport systems. Individuals that have liver disease do not excrete the manganese because most of the manganese is excreted through the bile. There is information from Bangladesh and other countries, and it's of concern in the United States because a lot of people get well water, and it has been shown that children that consume water that has levels of manganese in excess of 300 micrograms per liter develop neurocognitive deficits. And then there's some other environmental exposures. MMT just reflects the fact that we're pretty good students of our history. We used to put lead as an anti-knock agent. We've taken it out because we know what price we paid for it. Now we're doing the same thing with manganese, and where it's being used, not in the US, but in other countries, it's been shown already that atmospheric levels of manganese have increased by at least twofold. And of course, if one is exposed to it from birth throughout senescence, there's heightened risk for elevated exposures. Welding is another risk. There are a number of studies showing that welders because they inhale a lot of manganese-containing fumes, develop Parkinson's disease at an age which is about 16, 17 years younger than the population at large. And the last one on the list here is a sort of endemic that's spreading through the U.S. It started in Russia, in East European countries, and its consumption, it's very analogous to the MPTP story, which some of you may be familiar with from the Bay Area, San Francisco, about 20 years ago. These are individuals that are consuming this designer drug, and basically the manganese is just a byproduct because it's used, potassium permanganate is used for oxidizing ephedrine and pseudoephedrine to the methcathinone. So these are the major exposure routes. Now there have been early studies actually that suggest that there might be a relationship between manganese levels in different brain regions and Parkinsonian dementia. This is from 1972. Areas that are affected in Parkinson's disease are basically the basal ganglia, and you can see that in Parkinson's disease cases, which is shown in white, the levels of manganese are significantly increased in PD cases compared to the controls. This obviously doesn't establish causation, but it's quite interesting. Now, a little bit of neuroscience, and the reason for the similarity between Parkinson's disease, idiopathic Parkinson's disease, and manganism, which results from exposure to high levels of manganese, is that these are basically the pathways that are affected in Parkinson's disease. So everybody's familiar with the substantia nigra and the striatum. These are the areas where you have neurodegeneration in Parkinson's disease. But then you have some projections with the globus pallidus and a projection back with the thalamus to the cortex, and another one from the globus pallidus or the subthalamic nucleus to the substantia nigra. So in Parkinson's disease, idiopathic Parkinson's disease, or familiar Parkinson's disease, this is the area that's been affected. Most of the manganese actually accumulates in the globus pallidus, but the way to look at this is basically look at it as a hose. It really doesn't matter where you block the flow of the water, whether it's here, or here, or here. Ultimately, water is not going to be coming out of the end of the hose, and that's why the symptoms of manganese are quite similar. So our lab has focused, as Charlie mentioned before, on different transporters for manganese. And there's a number of importers, which are shown here. I'm not going to go through all of them because there's not enough time. I'm going to focus on the DMT1, which is a divalent metal transporter. It's not a very specific transporter. It transports metals in the divalent oxidation state. It does transport iron. Hopefully I'll convince you that it transports manganese. There's another iron transporter in the 3+ oxidation state, which is by way of the transferrin receptor. And although manganese in the 3+ oxidation state is a very small amount, it's found in a very minute amount in the blood, I'll show you some data hopefully convincing you that manganese can be transported on the transferrin receptor. I'll start by characterizing a transporter for manganese, an exporter, which is ferroportin, which is the main exporter for iron. And I'll discuss some recent studies that have been published on what was thought to be a zinc transporter, and we actually think it's also a manganese exporter. So let me start with ferroportin. So the question that we ask here basically is whether ferroportin is a transporter for manganese. And what we've used here is HEC-293 cells with a ferroportin construct that's hooked up to green fluorescent protein. And this cell line has two introduced elements. One of them is a stably expressed ecdysone receptor and an ecdysone-inducible promoter, which is upstream of the recombinant ferroportin gene, which is fused with a GFP tag. So in order to activate the expression of ferroportin in these cells, you basically have to use an ecdysone analog. And I think this will be clear in the next slide. And the analog that we use is phonasterone A. So these are the HEC-293 cells. You basically don't see any green fluorescent protein fluorescence. If you transfect the cells with this complex or construct and you do not use the phonasterone, they look the same. There's no expression of ferroportin. And if you transfect them with a construct and you use pronasterone A, you get green fluorescence. And this is also recapitulated here in a Western blot. So basically, no band for the ferroportin. These are the control cells. No band for the cells that are transfected with the construct. And we do not use the pronasterone. And this is the ferroportin band in the cells where we are transfecting them and using pronasterone. We're using the pronasterone to induce the expression of ferroportin. So some crude measurements on toxicity of manganese in these cells. Here we're looking at control cells at different concentrations of manganese, and these are physiologically relevant concentrations, up to about 150, 175 micromolar. If you go above those levels, these are levels that you'd find in mammalian systems. As you increase the concentration of manganese in HEC293 cells, you see an increase in the release of the lactate dehydrogenase, which can be used as a measure of toxicity. It usually is retained pretty well within the cell. If you induce cell damage, specifically to the membrane, the LDH can be recovered within the medium. So this is basically what we do here. And as you increase the concentration, you see you get an increase, significant increase in the amount of LDH in the medium. When we transfect the cell with the construct and we use the pronasterone, you can see pretty much that for each of these concentrations that we use, the level of LDH that we find in the extracellular media is significantly reduced in the cells that are overexpressing the ferroportin, suggesting that having the ferroportin in these cells is protective against manganese. Another assay that one can do is basically a mirror image. We're looking at MTT. I'm sure most of you are familiar with this. It's a measure of mitochondrial function. Again, as you increase the concentration of manganese, you see a reduction in mitochondrial viability. And if you transfect the cells with ferroportin, you induce it with a pronasterone. For each of these concentrations, again, the level of damage, mitochondrial damage, seems to be attenuated in the cells. So the next obvious thing to do was to measure how much manganese is in these cells. This is shown here. This is after 6 hours exposure. Here we have all three sets of cells. The HEC293 cells, the ones that have been transfected with the construct, but in the absence of the pronasterone, and the cells that have been transfected in the presence of the pronasterone. And you can see in these two, the levels of manganese are pretty much the same, and the levels of manganese with the pronasterone induced cells is significantly decreased, again consistent with the damage that we see in terms of the LDH assay and the MTT assay, and suggesting that the ferroportin is likely a transporter for manganese. We've also looked at the relationship between manganese and iron using MRI. This was done at Vanderbilt. I'll just show you a couple of slides. The nice thing about manganese is that it's a paramagnetic metal, so you can use MRI to look at the accumulation of the metal within the brain. And what was done here basically was to inject rats with 3 milligrams of manganese per kilogram for about 14 weeks. And we aimed to reach concentrations of manganese in the blood of 6 to 12 micrograms per liter. 12 micrograms per liter. The 6 to 12 is about normal levels. And these are levels that recapitulate manganese blood levels in welders, for example, and other people that may have been exposed to manganese. The beauty of the MRI is that you can obtain an initial scan of each of the animals, and then you can follow them all the way through 14 weeks. And we looked at these animals every 2 weeks. And this is basically what you see. So this is a low Tesla resolution, so the resolution is not great, but I think you can appreciate that in the treated animals, and these are coronal sections, and in sagittal sections, there's much more manganese in the manganese-treated animals. And the manganese accumulates in various regions. A couple of them of importance are the areas that I mentioned before, the basal ganglia. You see more manganese in the cerebellum. And one of the interesting findings, actually, although these animals received the manganese by way of the intravenous injection, there was a lot of manganese within the olfactory area, And the reason for that is that the olfactory bulb is known to have very high level density of the divalent metal transporter 1. So although it was given into the blood, because of the DMT1 within this region, a lot of the manganese was localized within the olfactory epithelium. We can then do some composites on these kinds of images, and I'll show it to you in the next slide. We can generate different brain templates for the controls and for the composites. We can set a statistical threshold at whatever value we want, 0.01 or 0.05, and where there's a difference in the level of manganese between the control animals and the treated animals, we can just put basically a red dot. So it doesn't give us an absolute quantity of manganese within the region. It just tells us that there's a statistical significance between control and treated animals. And this is basically the way it looks. So it's symmetric. You see it on both sides. You see it pretty much throughout the brain, but it's very prevalent in those areas that I mentioned before. These are animals that receive the same amount of manganese, okay, to these animals, but what we've done here is we've put them on, I think it was 6 weeks of iron depletion. And again, without any statistics, I think you can appreciate that in animals that are iron depleted, the amount of manganese that's in the brain is significantly increased. So being iron depleted, which is a global problem, there's about 1 billion people around the globe that are iron deficient, increases the levels of manganese within the brain. Okay, next I'm going to talk about the transferrin receptor. This is some work that's been done with Tom Gunter at the University of Rochester. And as I said before, there's very little manganese in the 3+ oxidation state, but it's a very strong pro-oxidant. So there's always been concern that it may actually be the species of manganese which is causing most of the damage within the various brain regions where manganese accumulates. So the question that we asked here was whether manganese can be transported in the 3+ oxidation state. This was done in tissue culture, so here we're using striatal neurons. And what we do is we take manganese in the 3+ oxidation state, we label it with Alexa green and with transferrin, and then we add it to the tissue culture, and I'm showing you just 2 points. This is a time lapse showing manganese in these cells at 2 minutes and 16 minutes. So the Alexa green is the manganese, and the red that you see is just mitotracker red to label the mitochondria. Now, if manganese was entering the cells, you'd expect to see a merge between the green and the red, so the color should be more yellowish or orange over time. So this is in 2 minutes. I think you can see the nucleus, but it's not very well delineated. You can see the nucleus much better delineated at 16 minutes. I think you can appreciate that there's less green at 16 minutes compared to the 2 minutes, suggesting that some of the manganese is moving in. We've done some kinetics as well. So here we're looking at manganese uptake, nanograms per milligram protein over time, up to 12 hours. This curve here is for manganese 2+, so it gets in much more readily than the manganese 3+, but nevertheless, you see that the manganese 3+ also has a positive slope, suggesting that it gets in. I mentioned that iron is transported on transferrin as well. So one way that you can ask questions about the transport of manganese is by mere competition with excess iron. So here we're looking at the transport of manganese, 3+, again, using Alexa green as a measure. And down here we're looking at the transport of the same amount of manganese, but we add excess of transferrin, 80 micrometers of transferrin, that 80 micromolar of iron that's bound to transferrin. And you can see that you can completely block the uptake of manganese that's hooked up to the Alexa green. You can also use some specific inhibitors of what's known as the clathrin-coated pit mechanism, which characterizes the transferrin uptake. And both of these compounds, chlorpromazine and dinosaur, again, completely inhibit the uptake of manganese in the 3+ oxidation state. So we think that the transferrin announcement. I'm going to switch. Okay, so as Charlie mentioned, the last 8-9 years, we've started to use C. elegans to look at different transporters and with mercury, with manganese, and some other compounds. And I'll show you some of the data that we've generated over the last few years. So some of you know about C. elegans. I have just one slide as an introduction. They're quite easy to culture. Obviously, one of the good reasons to use them is the power of genetics. For those interested in the nervous system, they have 302 neurons compared to a few billions that we have. As far as dopaminergic neurons, which were mentioned before, they have 8 of them. So they're very easy to look at and count. The first question that we asked was about manganese transport. And we decided to look at DMT1. I don't expect you to see much here. This is just basically genetic or sequence homology of the DMT1 all the way from yeast up to humans. C. elegans is right here. And where the color is light blue, there is fairly good homology between all these species. Now, to make things a little bit more complicated, actually, DMT1 in C. elegans is referred to as SMF1, SMF2, and SMF3. So there's 3 different isoforms of DMT1. And the question that Catherine and Alex asked in the lab was, what's the role of DMT1 in the transport of manganese? And if we knock out DMT1, are the animals going to be more resistant to the toxicity of manganese? And this is just a survival assay. This is the log dose of manganese. Those that can do the calculation pretty fast will see that the levels are pretty high. The dose is pretty high. The reason for that is that they have a very thick cuticle, so you need to use fairly high concentrations. Our prediction was that if we knock out the different DMT1 isoforms, that the animals would be less sensitive to manganese. So there would be a whiteboardly shift in the dose response curve. And you see that actually with 2 of the DMT1 knockouts, the SMF1 and the SMF3. But to our surprise, what we found out was that the SMF2s were more susceptible to the manganese. We measured the manganese concentrations, and SMF2 knockouts actually had higher levels of manganese compared to the controls, which is consistent with what you'd expect. So what Alex and Catherine went on to do was to use transcriptional and translational GFP fusions to localize where the transporters are so that we can come up with a model for the transport of manganese in the worm. And I'll show you the next 3 slides basically just have each a summary of where the transporter is. These are the different images. So most of the SMF1, which is shown in blue, is found within the gut. SMF2, which is in the next slide, is localized in the pharynx, in this area here. We couldn't find anything in other regions, or very little. And SMF3 is very analogous in its distribution to SMF1. So there's a consistency between what I've shown you before in terms of the sensitivity to manganese and the viability data and the distribution of these different isoforms. We've gone on to do a number of other studies, and I'm not going to show you data, but basically this is the working model that we came up with, which suggests that the SMF1 and SMF3, shown in blue and red respectively, are basically the homologs of the DMT1 in mammalian system, whereas the SMF2, which is in the pharynx, is more of a sensing and uptake regulation mechanism for inhibition of the uptake by way of pharyngeal pumping. The next transporter is SLC3810, which I mentioned was thought to be a zinc transporter. A couple of years ago, we came across a paper that suggested that humans that have mutations, and a couple of them are shown here, a sense and a missense, a nonsense and a missense here and here. These individuals tend to develop Parkinson's disease, and the thing that struck us was that these individuals have high levels of manganese in the blood. So we thought perhaps this is a transporter for manganese. It's also localized in the brain. It's a 6-membrane domain, as you can see here, transporter, and I've already mentioned the mutations. So the hypothesis was basically that both in worms and other systems, this is a manganese transporter. It would be expressed in dopaminergic neurons, and the wild-type transporter would be functional, hence we see it docked within the membrane, whereas the mutated forms, L89P and other mutated forms, will lose their activity and therefore would be localized within the cytoplasm. The first thing that we did with collaborators at the University of Texas in Austin is look at the SLC3810, where it's distributed in HeLa cells. So this is SLC3810. This is a wild type. Calnexin was just used as an integral protein of the endoplasmic reticulum. This is a merge. And these are different mutations in the SLC3810, and I think you can appreciate that the mutated form is very differently distributed from the wild type. It's mostly in the cytoplasm, and actually very little of it, if any, is found within the membrane. We've done some bioaccumulation studies, which are shown here. You can see that we find the SLC3810 within the membrane. We do not find the mutated form within the membrane, and we used another protein, GPP130, which I'll talk about in just a minute, which we know is not found within the membrane, cytoplasmic membrane, and again, if you do bioaccumulation assays, you don't find any of it within the membrane. So most of the wild-type transporter is in the membrane, and hardly any of the mutated forms of the transporter are in the cytoplasmic membrane. Now, the protein that I'm going to mention is GPP130. Very briefly, what it does is when manganese concentrations in the cell are elevated, GPP is a chaperone protein. So what it does, it chaperones the manganese from the Golgi to endosomes and lysosomes, where the GPP is degraded. So these are control cells with GPP, red here. You can see when you add high levels of manganese, the GPP130 completely disappears. The reason it disappears is because it shuttles the manganese to the lysosome and to the endosomes. Now, if you do the same experiment and you transfect the HeLa cells with SLC3810 and treat with the same concentrations of manganese, you can preserve the GPP130, essentially because now you don't need the GPP130 to shuttle the manganese to the lysosomes and endosomes. You have a very efficient mechanism for the removal of manganese from the cell by way of the SLC3810. This is the same thing. So this is again a transfection with the SLC3810. You add the manganese, you see high level of expression of GPP130, but if you do it with the mutated forms of SLC3810, the GPP disappears, suggesting that the transporter is now dysfunctional. We've looked at the intracellular concentrations of manganese to validate what I've shown you before. And I'm just going to show you this panel. So here the concentrations of manganese are back to the micromolar range. These are concentrations, part per billion microgram of the lysate. These are the control cells. And if you transfect the cells with a wild-type SLC3810, the amount of manganese in these cells is significantly reduced. We've also looked at viability, which is shown here. 100% with no manganese. As you increase the concentration of manganese, viability decreases. Upon transfection with a wild-type SLC3810, viability is higher, significantly higher compared to the controls. And we've gone on to characterize primary midbrain neurons to look at neurodegeneration in different mutants and in wild-type. Just very briefly, we're looking at primary neurites in wild-type and mutated forms of the transporter. We're looking at total number of primary neurites, total number of neurites per neuron, and so forth. And you can see that in wild-type cells, if we have the manganese, there's no difference compared to wild-type in the absence of manganese. cells with a mutated form of the transporter. In each of these cases, there's a significant change in each of these endpoints of toxicity. So in the mutants, ones that express the mutated form of the transporter, there seems to be much greater neurodegeneration compared to cells which are transfected with a wild-type SLC3810 transporter. This was carried on by Pan, who's here in the audience somewhere, over there, to look at the potential role of SLC3810 also in manganese transport in the worms. What we did first is to look for homologs of the manganese transporter of the SLC3810 in worms, and we found a number of them, but you can see the homology was very low. So our conclusion was basically that the worm's body do not express the SLC3810. Penn did some RNAi studies where he knocked out these different genes and looked at the viability in the presence of manganese, and they couldn't really find anything. If there was any significant change, actually, with knockdown on one of the transporters or the genes, it seemed that the worms actually had higher viability compared to the controls. So rather than do this and focus on these transporters or genes, what we decided to do is express the human SLC3810 in the worms. So we have three different groups here. We have the control worms, we have controls, we have worms where we express the wild type, SLC3810, the human transporter, and this is the transporter that I've shown you before, which has been associated with Parkinsonism, the L89P. In the absence of manganese, there's no difference in viability between these three different groups. If we add manganese at different concentrations, there's a significant decrease in the viability in the controls. There's a significant decrease in the L89, the mutant form, expressing worms, and we can rescue, significantly rescue, the ones that we transfect with the SLC3810. We've also looked specifically at dopaminergic neurodegeneration. So this is the head region, the nose would be here. These are 6 neurons that are found within the head. What you see here is GFP that's hooked up to the DAT promoter, the dopamine transporter. So only dopamine neurons express the green fluorescent protein. And you can see as you increase the concentration of manganese, you lose the axons, and as the concentration is further increased, you completely lose them and lose fluorescence even within the cell bodies. We have the same groups here again, the controls, the wild types, and the mutated form of the SLC3810. These are animals at a younger age, so the concentrations are slightly lower, and you see pretty much similar results. This is now focusing solely on dopaminergic neurodegeneration, a significant decrease with manganese in the controls, even further decrease in the mutated form of the transporter expressing worms, and some rescue with both doses in the animals that have been that are expressing the wild type SLC3810. We've also looked at the distribution of the transporter, both in the body wall muscle cells, which is shown here, and the dopaminergic neurons, which will be shown in the next slide. You can see again that the wild type SLC3810, analogous to what I've shown you in the HeLa cells, is predominantly expressed in the outer membranes, whereas the L89, the mutated form, is expressed throughout the cytoplasm. These are the dopaminergic neurons. You see it again, it's quite faint, but it looks very different compared to the mutated form. So from this, we basically concluded that the worms do not appear to express this transporter. The wild type SLC3810 attenuates the toxicity of manganese. The L89P does not. This form of the transporter doesn't seem to dock within the membrane, so it's likely dysfunctional, whereas the SLC3810, the wild type, both in dopaminergic neurons, in HeLa cells, and in HeLa cells seems to be functional because it's primarily expressed within the cytoplasmic membrane. Now the machinery for dopamine synthesis is intact within the worm. They have the full machinery that we find in mammalian systems. There are some differences in terms of nomenclature again. I'm not going to go through the whole process. I'll just show you a couple slides. Basically, we can query the reason for the sensitivity of dopaminergic neurons to manganese in this species. One of the species that we looked at is animals with a knockdown of tyrosine hydroxylase. Tyrosine hydroxylase being the enzyme, rate-limiting enzyme for dopamine synthesis. And the others are animals where we've knocked down the dopamine transporter, which removes the dopamine from the synapse, from the extracellular fluid. This is a movie of these worms. So again, you're looking here at worms where the dopamine transport, DAP1, has been labeled with a green fluorescent protein. You see 6 neurons in the head. I've shown you that before. You see a couple of neurons in the tail once in a while. So basically, you can add any compound that you might be interested in and probe the sensitivity of these neurons, or you can look at any other kind of neuron that you might be interested in. This is shown here. So this is similar to what I've shown you before. These are the dopaminergic neurons. There are strains where GFP labels GABAergic neurons, cholinergic neurons, chemosensory neurons, and so forth. And these are the controls. These are the manganese-treated worms. There are a couple worms here and a couple worms here, only one here and one here. But basically, the only difference upon manganese exposure is that the dopaminergic neurons seem to generate. There seems to be no effect on GABAergic neurons or cholinergic neurons or other neurons. So there's something that's peculiar to the dopaminergic neurons. So in a series of studies, we've looked at this sensitivity, and I guess, again, I'll just show you a few slides. Here we generated the dopamine transporter knockouts, the CAT2 knockouts, and generated a double mutant of dopamine transporter and tyrosine hydroxylase, CAT2 knockout. And it's interesting because when we knock out the DAP1, actually, we find that the animals seem to be more sensitive to manganese. There's a leftwardly shift in the sensitivity, which suggests that it might be something about extracellular dopamine, actually, that makes these animals more sensitive to manganese. If you knock out tyrosine hydroxylase, CAT2, the animals are less sensitive because they don't make any dopamine. And if you make a double mutant, the double mutant animals look like the CAT2 mutants. It's really irrelevant that they don't express the DAP1 because in the first place they don't make any dopamine. So there's no function for the dopamine transport. If you add exogenous dopamine to these animals, they become more sensitive. Again, there's a leftward shift. You can measure dopamine in these animals. You can do a lot of other assays. I'm not going to go into that, but basically the finding is that dopamine is essential for manganese toxicity. It was mentioned, Charlie mentioned that we have an interest in oxidative stress. This is shown here. The major switch for antioxidant responses in cells is via the NRF2 mechanism, which in C. elegans is known as SKIN1, SKN1. Basically, when you have electrophiles, the reactive oxygen species, you get a couple cysteines on keep1, which is usually associated with NRF2 to oxidize. This results in the dissociation of the keep1 from the NRF2. NRF2 moves into the nucleus, it binds to an antioxidant response element, and you get upregulation of all kinds of antioxidant stress protein, phase 2 detoxifying enzymes, and so forth. We've generated some SKIN1 GFP tagged animals and some knockouts of SKIN1. And this is just to show that if you look at animals that do not express the same levels of SKN1, 25% reduction, they seem to be much more sensitive to manganese, whereas animals that overexpress SKN1 seem to be more resistant to manganese. So basically, this is the model that we came up from all these studies. Manganese is transported in the 2+ oxidation state on SMF1 and 3. As I mentioned, SMF2 seems to be a sensor for manganese. I've shown you some data to suggest that if you knock out the dopamine transporter, there's increased sensitivity to manganese, suggesting that there's something about extracellular dopamine that probably is responsible for this sensitivity. SKN1 is important, again, suggesting that oxidative stress is one way of mediating dopaminergic cell death in the various animal models. And I've shown you earlier some of the transporters. We think that SLC3810 is a transporter or exporter for manganese. Ferroportin is an exporter for manganese, and that manganese can be transported in the 3+ oxidation state on transferrin. I'm going to finish up, well, this basically summarizes what I've said. I'm going to finish up by talking for a few minutes about parenteral nutrition. So parenteral nutrition has been around for quite a while. You can see it's been used in infants for about 47 years. And it's added to parenteral nutrition basically as a supplement without really very much scientific basis. Initially, in '85, the American Academy of Pediatrics came up with a dose of 2 to 10 micrograms per kilogram per day. And this was revised in 2003 by the American Society for Parenteral and Enteral Nutrition, and it was reduced from 2 to 10 to 1 microgram per kilogram per day. But as I said, the scientific basis for this is lacking. There is really no basis for it. So what we've done, we've looked at manganese concentrations in various neonatal and nutritional formulas. And the first one is the best, and it's human milk. And you can see the manganese content. Now, as I mentioned before, this is an essential element. Actually, in neonates, the absorption is higher. It's about 8%. So babies consuming maternal milk will absorb about 8% of the dose. Babies on cow formula get more on a liter basis, but there's a slight reduction in the absorption, compensating for the difference. And soy formula actually has a lot more manganese, but the absorption is very low, again, compensating to a certain extent for a different amount. Now, babies that receive parenteral nutrition without trace element supplements actually get 7 micrograms per liter. But remember, they get everything IV, so 100% of it is absorbed. And the multi-trace 4 neonatal formula is used in the United States and many other countries. And babies getting this formula get about 2,500 per day, micrograms per liter. And the absorption is 100%. Now, a lot of these babies are neonates. They're premature. Their blood-brain barrier is not developed. Their biliary tree is not developed. So manganese very likely more readily gets into the brain. Excretion of manganese is not functional to 100%. And if you actually look at how much these babies get, this is human milk, cow milk, soy formula, parenteral nutrition, this is in the absence of the trace element supplement. And these are the babies with a trace element supplement. We see adult toxicity at about 2 micrograms per kilogram per day. And these babies get a lot more. Actually, if you do some calculations, it turns out they get about 100 times more than they would get if they were just nursing from their mother. So the overall goals of this project was to look at neonatal populations and increase risk for excessive brain manganese deposition in the brain by using MRI. Again, it can be used because manganese is paramagnetic. And look at the relationship between manganese deposition and dietary manganese intake, the degree of prematurity, and liver function. The hypothesis was that there's a direct association between shorter T1 relaxation time. Shorter T1 relaxation time means more manganese in the brain. Okay, there's an inverse relationship between the T1 weighted relaxation time and the accumulation of manganese. This was done in the globus pallidus because it's an area that we know manganese accumulates, as I've shown you earlier. And we looked at the MRI images in relationship to total dietary manganese, total days of parenteral nutrition, the liver function by looking at conjugated bilirubin levels, and blood manganese concentrations. These are some of the methods. So MRI was done. We measured manganese levels in the blood by using ICP-MS. We had fairly good, or extensive actually, dietary logs on how much parenteral nutrition these babies received from birth to the date of the MRI. And the statistical method was by multivariable linear regression. These are the two groups of babies that we had. The NICU parenteral nutrition group, which received manganese for at least 4 weeks prior to the study, and greater than 75% of their nutrition was by way of parenteral nutrition. They were greater than 30 days of postnatal age, and obviously they had to be stable enough so they could be transported for the MRI. You cannot do this kind of study in control babies because you can't really do any procedure on babies unless it's beneficial for them, and MRI is not beneficial. So the control groups are babies that were in need of a clinically indicated MRI. They were less than 2%, less than 2 weeks on parenteral nutrition, and obviously it was important that they had no liver disease. So these are the study populations. There are a couple groups here, the NICU parenteral nutrition group and the control group. As you'd predict, the NICU group are younger. There's statistical significance. Gestational age, the gestational period in humans is 40 weeks. So you can see these babies are quite premature. The MRI was done at the same time, post-menstrual age. The MRI is the same, which is important. The total number of days on parenteral nutrition prior to the MRI is significantly different between the two groups, and the babies on the parenteral nutrition received about 9 times more manganese compared to the control group. This just shows the relationship between gestational age and T1R relaxation time. This is the gestational age at birth versus the T1 relaxation time. Remember, a negative slope or shortening of the T1 wave indicates greater concentrations of manganese in the brain. So at 40 weeks, you can see the babies have more manganese in the brain compared to the babies that are born at 25 weeks. If you look at these images, and this is not the best image that I could find, I actually think if I gave you 10 of these images from control babies and the parenteral nutrition babies, that 9 out of 10 cases you'd be able to classify into the correct group because it's pretty striking. These are the basal ganglia. You can see a lot of the white density reflecting the manganese in the NICU parenteral nutrition group, and you can't see any of it in the control group. This slide shows the association between the dietary manganese and the T1R, and we broke it down to different gestational ages. So if you actually fix the point at, let's say, 200 micrograms of manganese, dietary manganese, you can see that gestational age 40, the babies have more manganese compared to the gestational age 24, consistent with what I've shown you in the previous 2 slides. Again, shortening of the T1R reflects higher levels of manganese. And then if you look within each of these groups, you can see that as the total dietary manganese increases, the level of manganese in the brain, or the T1 relaxation time, also increases. So there's more manganese at 1,000 micrograms total dietary manganese compared to 200. And you see throughout the different gestational ages. These are the whole blood manganese levels in these babies. The normal is 8 to 12 micrograms per liter, and the babies on parenteral nutrition had blood levels of 60 micrograms per liter. So it's about 10 times higher compared to the control levels. And if you look at liver function and manganese accumulation, this is a measure of liver function, conjugated bilirubin. The higher the number, the more dysfunctional the liver. Again, shortening of the T1 relaxation time within each of these groups as parenteral manganese increases. And if we compare the two groups, or the three groups, you could see that if we fix at any of these points, let's go back to 200 micrograms in the more dysfunctional liver, 6 conjugated bilirubin of 6, there's more manganese compared to the conjugated bilirubin at 2. So again, liver function seems to be a risk for increased deposition of manganese in the brain. So dietary manganese exposure is inversely associated with relaxation time in the globus pallidus, T1 relaxation time. T1 weighted MRI can be used to screen infants on prolonged parenteral nutrition for increased deposition of manganese in the brain. Liver disease is a risk factor for increased manganese deposition in babies that get fed by way of parenteral nutrition. We think the trace element additives must be reformulated. We think there's way too much manganese in parenteral nutrition. And obviously, the question is, what are the consequences of this treatment or clinical practice where the babies get a lot of manganese in their parenteral nutrition? And this is just sort of a hypothetical slide. This is my last slide. I see it's still 2:15. Okay. So let's say that you need to lose 40% of dopaminergic neurons in order to develop symptoms that show that you clinically have Parkinson's disease. It's actually more 90 or 85%. You can see that as we all age, we lose neurons. We are going to hit this hypothetical line, normal decline with age, at about 150 years of age, so it's irrelevant, the same at 0.1%. However, if there is a decline in the number of the cells at 0.5% or 1%, it becomes significant. Then one may develop Parkinson's at a much younger age. So the argument would be that in these babies, what we may be doing is we may just knock down 20 or 30% of the dopaminergic neurons. So their complement is at about 70%, let's say, at 25 years of age. Even if you extrapolate then this line, and it's parallel to normal decline with age, you can see that these babies are going to reach this imaginary line at a much younger age. So the real question, and there's no, people always ask me this question, that there's no epidemiological data. But the question is, are we setting up those people, those babies, to develop neurodegenerative diseases, specifically Parkinson's disease, at a much younger age? And I'll stop here. These are some of the people that did the work, the ones mostly in the yellow. I'm not going to go through all of them. Ken is here, and in the audience who did the C. elegans work. The transferrin work was done with Tom and Carlene Gunter. Folks here did the work on the babies, along with some people in the Center for Imaging. And Samush Bara, Mukhabadai, did the work on the HeLa cells that I've shown you with SLC30A10. Last but not least, some of the funding for this. Actually, the Gerber Foundation was the first one to fund studying the neonates, and then we had, at the time it was still called the GCRC. We had some money from Vanderbilt, GCRC to do it, and most of the other work was funded by the NIEHS. And that's it. That's a picture of myself. Stop here, and I'm happy to take questions. Yes?
You talked about like almost an inverse relation between iron levels and manganese levels. Are there any clinical correlations to that? For example, in populations that have iron overload, hemochromatosis, is there to be a lesser sensitivity to manganese, or the inverse, is there iron deficiency?
Yeah, so there are no clinical relationships. And we've also actually looked at the relationship between iron repletion and the uptake of manganese into the brain. We thought could be very smart by just giving animals some extra iron to show that their manganese levels go down, but it doesn't work this way. Actually, animals, even animals that are on iron repletion, show high levels of manganese in the brain. I think the reason for that is that the little manganese that gets into the cells is pretty much trapped within the cells because the transferrin receptor has a much higher affinity for the iron, for the export. So when you have excess of iron within the cell, the manganese just can't get out, basically. Iron deficiency is more prevalent also in females than in males, but we know that Parkinson's is more prevalent in males than in females.